All right, so what we're going to do now is we're going to keep talking about navigation, but we're going to move beyond these regions. And one of the first regions we're going to move into is actually the hippocampus, because the hippocampus is really one of the dominant links between navigation in the brain, specifically in humans and as well as a lot of other types of animals. And just to orient you again, the hippocampus is this subcortical structure which is kind of deep in the brain, in the medial temporal lobe. So here's the thalamus, hypothalamus, and amygdala. The hippocampus is deep in there. And so if you take this sort of three-dimensional view of it, you can kind of see it. It's these two little wedges or slices that are there in the temporal lobe, but are on the inside of it, hence medial. And to really get a sense of where the hippocampus is, it's actually probably easiest when you just get a cross section like this, because here is the corpus callosum, and then these two little slivers down here, this is the bilateral hippocampus in humans. And just to give you a sense of what it looks like, this is the big bilateral hippocampus in rats. And this is actually something worth pointing out, because a lot of the way people have learned about the role of the hippocampus in navigation actually initially comes from studies of the rat hippocampus. And so the way these studies usually work is by having these sort of electrode arrays implanted chronically in a skull. And actually what you can do is you can implant them chronically in this way so that there are these wires that are attached so that the animal can freely navigate different sort of situations, and you can monitor what's going on in the hippocampus while the animal is moving around in a relatively normal way. And the first thing that was initially discovered in rats, but then was later discovered in humans, are these things in the hippocampus that are called place cells. Okay, now to get a sense of what a place cell is, what I'm going to first do is I'm going to show you a video, and I want you to kind of look at the response of this particular cell and see if you can kind of describe it in your head. Okay, so here is a rat. You can see its tail there. It's got a little electrode array implanted on it, and it's in a little 1 by 1 by 1 meter box, more or less. Might not be a perfect square. And the animal is going to be kind of freely running around and exploring this little box. And every time there's a red dot, that means that a cell in the hippocampus is firing. So right now, what we're going to be watching is a video recording of the firing pattern of one cell in the hippocampus that's going to be called a place cell. Watch the pattern of activity on the brain or on the screen and see if you can kind of get a sense of why we might call it a place cell. So here we go. So there's one fire. There's a bunch more. Now see if you can get a sense of the pattern. Probably by this point, a lot of you have kind of put together that this cell, while it might fire kind of randomly or sporadically out in these other parts of it, of the grid, it really seems to fire whenever the rat is in this one particular place. So if you make a heat map basically of the firing pattern, you see something like this, that this is a nice summary of that particular neuron's firing pattern. Whenever the animal is in that particular place, the neuron in the hippocampus will fire, and thus it's called a place cell, because it is a cell in the hippocampus that only fires in that one place. Okay, so that's one neuron in that one place, but what about all these other places in the grid, and what about all the other neurons in the hippocampus? Well, it actually turns out that there are a bunch of different place cells at any given time for a bunch of different locations. So for example, what we were just looking at was a place cell that kind of looked like, a situation like this. It was kind of on the lower left. Maybe it was closer looking like that one. But there would be place cells that are more associated with the upper right or the upper left or somewhere in the middle. So each one of these plots corresponds to the firing pattern of an individual cell. This is like cell 1, cell 2, cell 3, cell 4, and they each like different things. But the thing that they have in common is that they will all respond whenever the animal is in a particular place, okay? There's actually these beautiful studies where the rats will have to kind of navigate these little sort of pseudo mazes, and you can simultaneously record from multiple of these cells at the same time. And so what has been visualized here is that each one of these dots represents the firing pattern from a different cell. So when the animal is in this little spot, the purple cell, as it were, will fire. When the animal moves down here, now the cyan or turquoise, I don't really know what color that is, that cell will fire. When it moves back here, the red cell will fire. Now the orange cell will fire. And so basically, as the animal is moving around, there's all these little cells that are responding to differing degrees as a function of where in that particular place the animal finds itself, okay? So then a question that people wondered is, do humans have place cells, okay? But then the other question is, well, what method would you want to use to find place cells in the hippocampus of humans? Can't use EEG, obviously can't use TMS, can't even use fMRI because the fMRI doesn't have enough spatial resolution. What would you need to use? You would use human electrophysiology. And so in this case, though, rather than implant these electrodes that kind of sit on the surface of the brain, they had to find patients who had actually these chronically implanted deep electrodes that would drill down, as it were, kind of like an oil rig drilling into the middle of the cortex, that can then be recording responses from neurons that are in the hippocampus. Now, when you have patients who have these sort of chronically deep implanted electrodes in their hippocampus, you can't really have them go walking around. They're, epileptic patients, they've had pretty extreme neurosurgery. You can't ask them to get up and go wander around a maze very easily. That's kind of dangerous. That's a risk you might be willing to take with a rat, but you're not really willing to take it with a person. So how could you get them to do something navigational to potentially elicit a response from a place cell when you can't get them to actually get up and move around? The answer is you use virtual reality and virtual worlds. And so basically you have them on a little computer screen with a joystick or a mouse or whatever, kind of navigating through this sort of virtual little town where there's one storefront that's for Griffin games, there's another storefront for a kite shop, and then another one for water sports. And they're laid out all around in this tiny little world. And basically what you can have these participants do, these patients, is just kind of get their bearings about it. You can basically drop them in this little town and say like, hey, I want you to kind of get a sense of where everything is. And then you can just watch how these hippocampus cells are firing as they move around. And so here is sort of a grid map showing you where all these, 9 locations are. So in this case, there's 9 locations in this little town. Here are 9 locations in white. And the red is showing you the path where the participant is navigating in this virtual world. And you can look at the firing pattern and the number of fires per second, that's what hertz are, as the person is moving about different virtual locations. And so you can just see that whenever the person is in this location, the firing rate is the highest. There's a place cell here that is firing faster when the person is here than when they move a couple, virtual feet over to the left or to the right, and the firing rate drops much more. So sure enough, by finding place cells in rat hippocampus, scientists were able to identify place cells in human hippocampus, okay? Now, a question that I got asked a couple of years ago when I was teaching this course that I'd never really thought about and I didn't know the answer to, but I'm glad someone brought it up because I got to learn about this, was, well, Professor Cohen, what about animals that don't operate on a 2-dimensional map, okay? So when in a situation like this, our participant is operating largely on a 2D world. It's like a flat world in which they move around virtually. When I was talking about the rat studies, the rats were always navigating in a 2-dimensional world where they're kind of operating on this flat maze. But there are, of course, lots of animals that don't operate in this way. They operate on a 3D map. They're bugs, they're insects, they're birds, and then in this particular case, they are bats. And so there actually have been studies that have been done to put these electrode arrays on the skulls of freely flying bats that will then drill down and put these electrodes in bat hippocampus, not rat hippocampus, but bat hippocampus. And they will monitor this bat as it's freely flying around a constrained environment. And it turns out that there are, in fact, place cells in bat hippocampus that are 3-dimensional, okay? So they will actually be in a particular location of 3D space, not just a particular location in 2-dimensional space. So while rats, because they're primarily dealing with, flat environments, have place cells about that land on a 2D fold or a 2D map, the bats, as they're flying around, their place cells, which are operating in the same function, they fire whenever the bat is in a particular location, but it's a location in a 3-dimensional type space. So you can actually see that as the evolutionary demands and the navigational demands of the animal change, the place cell system in the hippocampus will change accordingly, okay? So is there any other evidence besides place cells showing that the hippocampus plays a big role in navigation? Is the only reason that we think of the hippocampus as playing a role in navigation coming from the existence of place cells? The answer to this question is definitely no. And this next experiment that I'm going to tell you about in humans is also one of my favorite studies probably in the literature, because it's an incredibly clever experiment using a kind of nuanced or idiosyncratic real world opportunity that actually you couldn't do in 2020. And I'll tell you why. So it used to be the case that if you wanted to be a London taxi driver, it was very, very hard, okay? Not just anyone could learn how to be a London taxi driver or could just sign up to be one, because the streets of London are very, very complex. The streets of London are not like the streets of New York. New York is in a large respect, except for, when you get downtown, a large respect is, horizontal, vertical by number. Whereas London is a very complicated city structure, so complicated that understanding how to get around London and to, get licensed as a taxi driver meant you had to acquire this thing that was referred to as the knowledge. It's really hard to learn it because back in the day, before we had GPS and smartphones, you couldn't just Google where you wanted to go and then follow, your GPS there. If someone was like, hey, I'm at this corner, I want to go to Buckinghamshire and Southampton, you'd have to know how to traverse this entire complicated thing to get to that particular point that you were trying to get to, okay? So it turns out that if you look at the size of the hippocampus volume, so HC stands for hippocampus volume, in London taxi drivers as a function of years of experience, you see a nice linear relationship that looks like this. That as people are spending more and more time driving a taxi cab around London, their hippocampus grows and grows and grows. And so you can see actually in this pretty crude measurement, we're just literally measuring how big is a person's hippocampus, and each one of these dots is an individual person, that the longer people have been driving their cabs, the larger their hippocampus is relative to people who haven't been driving it as long, okay? And then it actually turns out that in these taxi cab drivers, people who have damage to their hippocampus, so you can see a little bit of damage to the hippocampus here, they lose their ability to use the knowledge to get around London. So in this particular case here, the red line is showing you the route that this driver could have or should have taken. They were at, St. Paul's Cathedral, and I think that's the Bank of London. They could, this is pretty straightforward actually. But in this particular guy, who was a London taxi driver, who was, quite a good one, when he was told to go from St. Paul's Cathedral to the Bank of London, he got very lost. He made a U-turn, he came over here, he drove around in a little circle, it wasn't until they got over here, where they got to, Kingsway and Holbrook or something? I don't know. When they got way over here, that they're like, all right, we're going to stop the experiment because he clearly has lost his capacity to navigate the streets of London, even though he already had them. So in this particular case, it's actually interesting because this guy didn't have generalized amnesia, which we're going to talk about in a little bit when we talk about memory and HM and long-term memory disorders. He seemed to be damaging a posterior part of the hippocampus, which seems to be uniquely involved in navigation. Sorry for the animation. So the last thing about these taxi drivers that I think is so interesting is that it turns out that some people just can't acquire the knowledge. I am probably one of those people. If you remember from the earlier video, I said I have a terrible sense of direction. I see a map like the streets of London and I'm just like, I give up. I don't want to do this. Please take me to New York. This is just, I'm always going to be lost. I bet you anything I'm never going to be able to acquire this. And it turns out that this is partly true, that there are some people who come in and they're able to learn the knowledge and they pass the test and they become taxi drivers. But there's some people who, even after a lot of experience, they're never able to do it, okay? So what we're going to look at here on the vertical axis is gray matter intensity, which is basically just saying how big is the gray matter associated with the hippocampus. And we're going to look at it across 2 time points. This could be 6 months. This could be a year. What is it? In my notes. Oh, it's 4 years later. Sorry. So time point 1 is at some point, and then time point 2 is 4 years later. And so here are control people who do nothing about the knowledge. You bring someone in, you measure their hippocampus, you bring them back 4 years later. If they're young enough and healthy enough, their hippocampus doesn't change in its size whatsoever. What do you think is going to happen between time 1 and time 2? In qualified trainees, you're going to see that their hippocampus gets much, much larger. The idea being that this part of the hippocampus that's involved in navigation will grow as their understanding of their navigational world grows accordingly. Whereas people who are non-qualified trainees and simply are unable to do this, part of the reason that they're unable to do this is because for whatever reason, their brains don't seem to have the flexibility or the plasticity to acquire this new knowledge. Why is this the case? Because there's individual differences. And you can see that the hippocampus literally grows as your knowledge of the knowledge grows and is learned. But if you're one of these people who just seems like you can't do it, part of it might be because your brain is not plastic enough. why I always get asked why, Professor Cohen, is it the case that there are these individual differences? Think about it, there's individual differences at everything. I bet, let's think of a sport. This is weird. Growing up, I played very competitive golf. I lived in the South. Everyone played, not everyone. A lot of people played golf in the South. It's a long story. My dad was a golf nut, so he kind of dragged me into it. I bet if I took a bunch of you who have probably never played golf, some of you could get a lot better at it than others. Why? Just because of natural dispositions, natural learning capabilities. Some of you might be more mathematically inclined than others. Some of you might be more, English, literature, prosaically inclined than others. Your minds just work that way. You make connections that some others could do. We could do art lessons, and some of you are going to get much better at painting than others. So there's just natural variation in what people can do. And so the point I'm just trying to make here is that you can see this natural variation literally manifesting itself in the size of the hippocampus, such that you can literally track how the hippocampus grows as your knowledge of the knowledge grows and is learned. Okay, so we've talked about the PPA, we've talked about the RSC, you're going to read about OPA, and we just talked about the hippocampus. It turns out that's not the end of it. We're going to do one more section on a few other regions and a few other different types of structures that are going to round out navigation, and then we will be done with that. We'll move on to memory, but let's take a break right there.